Hey everyone, welcome back to Window Seat Studies. From today onwards, we are going to learn about different histone post-translational modifications one by one in detail. So in the today's video lecture, we are going to learn about histone acetylation. In the previous video lecture, if you haven't watched it, we have discussed about all the different types of post-translational modifications that are done on histones, which we are going to be learning in detail and on which amino acid residue they are done. So today we'll learn about histone acetylation. But before learning about histone acetylation, let's learn about the consequences of those histone post-translational modification, or like what is the mode of functioning. Okay, what is the mode of functioning of those post-translational modifications, like what are the or you can say what are the consequences. So post-translational modification or histone are done for changing the chromatin environment, either making it densely packed or loosely packed. So it regulates the transcription by changing the chromatin environment. So the mode of functioning by so for changing the chromatin environment, there are two modes of functioning. First, you add directly perturb the directly so you directly perturb the chromatin architecture. So you can due to addition of certain chemical group, there will be a direct conformational change in histone, and that conformational change results in the alteration of DNA histone interaction. And this, so the whenever the protein changes, there will be conformational change in protein. It can change, it changes the that protein interactions, protein protein interaction. But here the histone conformational change changes the DNA histone interaction, and due to change in DNA histone interaction, the chromatin will be either loosely packed or densely packed. So the first mode of functioning is that you can directly perturb the chromatin, or other way is that you modulate the recruitment of the regulatory proteins. Okay, so like for example, polycom group of proteins. This is a group of proteins which helps in the reparation of certain gene. So those are recruited on the chromatin if the chromatin has certain mark. Okay, so those post transcriptional modification, those marks, those histone marks decide which kind of regulatory protein is going to recruit on that chromatin, and on that type of regulatory, on depending of that regulatory protein, the gene will either silence or activate. So this is how they work. Either they can directly perturb the chromatin, or they can recruit certain regulatory proteins. So these are the two modes of functioning. So now let's start learning about histone acetylation. So from the name, it's obvious that histone acetylation means addition of acetyl group on lysine. It is done on lysine residue. So it is done on lysine. So the acetyl group is added on lysine. So what happens is that whenever the acetyl group is added on the histone, so this is histone. Okay, and on this the acetyl group is added, so it gets, I'm just writing is it as this. So imagine, assume that it is histone 3, so it will be H3 acetylation. Okay, H3, and it's, well, if it's happening on the ninth lysine residue, so let me write it as, so I told you how to write it. So if it is happening on H3 on the ninth lysine residue, it will be written as like this. So this is the acetylation of histone. So what happens is that whenever acetyl group is added on the lysine of the histone, it neutralizes the positive charge of the histone. Why? Because see, lysine is the positively charged residue, So the addition of acetyl group, which is a negative group, it neutralizes the positive charge on the lysine. So what happens is that when DNA and histone interaction are all because they are all because of the charges. So histones are positively charged while DNA is negatively charged, and that is why DNA is wrapped up around histone. But due to neutralization of this positive charge, now the DNA histone interaction, it will be it will weaken up. Okay, so it is weakening the histone DNA interaction. So tell me what will happen. Will it make the chromatin more dense or loose? Pause the video, think about it, and what will be the answer? Whether it will pack it densely or loosely. So the answer is that whenever DNA histone interactions are weakening up, it is going to loosen up the DNA chromatin. Okay, it's an obvious answer because DNA histones are used for packaging of DNA. On that histones, the DNA is wrapped up and it is tightly packed. But now DNA and histone interaction is weakened up. So now the DNA, which is like a thread and which is wrapped up around the beads, which is histone, so it will now loosen up. So that loosening of DNA will make DNA more accessible. So now the DNA is more accessible. Okay. So now transcriptional machinery can come and read DNA and can transcribe the DNA, and this leads to the activation of genes. And that is why in the previous video lecture, I know that you must remember that histone acetylation is active mark. Why is it an active mark? Because it is neutralizing the positive charge, which is loosening up the chromatin, and this loosening of chromatin is making the DNA more accessible, and which is leading to the activation of gene, and that is why histone acetylation is considered as an active mark. So I told you that you have to remember which are the active mark and which are the repressive marks. So you can now always guess which will be the active mark. Because why? Because it is loosening up the chromatin, and that is why histone acetylation is an active mark. So here you can see the histone acetylation works through the direct perturbation of chromatin. Why? Because it is neutralizing the positive charge of histone. So it is making the change in histone and DNA interaction directly, and that is why we can say that the mode of functioning of histone acetylation out of those two is the direct perturbation. Okay, direct perturbation of chromatin. So this is it about histone acetylation. We will learn about the enzymes which are going to do the histone acetylation. Those enzymes are known as HATs, histone acetyl transferases. We will learn about HATs as well as the histone deacetylases in the next lectures. So till then, see you. Bye bye.